To convert the information stored in DNA into functional molecules such as RNA and proteins, a large amount of energy is required. Therefore, gene expression is strongly regulated. Via this regulation, the gene product activity of mainly proteins is also controlled. This enables cells to respond to environmental changes, for example, a change in the nutrient supply. The first fully described genetic regulatory mechanism is the lac operon in E. coli bacteria. Today, it still represents an adequate model for prokaryotic gene regulation. An operon is a transcription unit of genes whose products are required under identical circumstances, so it facilitates the coordinated expression of multiple genes. The DNA sequence of an operon comprises 3 different components: a promoter, an operator, and several genes, each of which codes for a protein. In the absence of lactose, a repressor protein is bound to the lac operator. This binding prevents transcription of the downstream lac genes. The repressor protein is encoded by the regulatory gene LAC-I. LAC-I isn't directly part of the lac operon, but is located a few base pairs upstream. As soon as lactose is present in the environment, it's taken up by the bacterium. Lactose then binds in the form of allolactase to the permanently expressed repressor protein. This binding inactivates the repressor, unblocking the operator. Now, the RNA polymerase can bind to the promoter and read the subsequent genes. This process is termed substrate induction, since it can only occur after the substrate lactose enters the cell. The 3 genes, LAC-Z, LAC-Y, and LAC-A, are now transcribed together as a polygenic mRNA. 3 different proteins are synthesized on this mRNA, namely beta-galactosidase, permease, and transacetylase. These proteins are essential to lactose metabolism. Permease forms pores in the bacterial cell membrane, facilitating further lactose uptake into the cell. The enzyme beta-galactosidase breaks down lactose to simple sugar residues that can then be metabolized. An additional special mechanism is used in the presence of lactose and absence of glucose. Glucose is usually the preferred energy source for E. coli. In the presence of glucose, lactose degradation is possible but not essential for survival and proceeds as just described. However, if there's a very low glucose concentration, the cell needs to break down as much lactose as possible. This is secured as follows. If little glucose is available in the cytosol, the CAMP concentration increases. CAMP then binds to the catabolite activator protein, in short, CAP. This CAMP-CAP complex forms a dimer that binds to the DNA close to the lac promoter, thereby increasing RNA polymerase activity. As a result, the 3 enzymes involved in lactose metabolism are synthesized at a higher rate, allowing the breakdown of more lactose, compensating the glucose deficiency. Through their concentration, both lactose and glucose affect the gene expression of enzymes involved in lactose metabolism. This enables rapid adaptation to different environmental conditions. In bacteria, genes are regulated by operons. Although the regulation of gene expression in eukaryotes is considerably more complex, it's based on the same concepts as, for example, the use of activator and repressor proteins.